Okay, thanks everyone for joining us. It is my distinct pleasure to have one of our first outside speaker in a while, and particularly having Dr. Matthew Lachniet here, visiting us from the University of Nevada, Las Vegas. So Matt is the director of the Stable Isotope Lab at Las Vegas, but he's also the chair of the department at UNLV, Department of Geosciences. And Matt, basically, I think I would describe him as a paleoclimatologist, maybe a speleoclimatologist. I like that term that Matt uses from time to time. And he specializes in trying to understand systematics of stable isotopes, but of several different types of isotopic systems, and using caves and carbonates to try and trace our paleoclimate history. Matt is an expert on climatology and climate change in the Pleistocene, particularly in the West, but his work has also taken him to the tropics. Particularly, he's an expert on Central American climate change over the Holocene and the Pleistocene as well. Matt started off as a PhD student working with Jeff Seltzer in Syracuse University. After he finished up there, he actually went and did a postdoc in Costa Rica. Is that right?
Panama.
Panama. I apologize. And then after Panama, he went and did a postdoc at the University of Massachusetts at Amherst. After that, Matt ended up joining UNLV. And Matt has won several awards. He was a Fulbright Fellow, I think, was part of this.
Fulbright to Costa Rica.
Fulbright to Costa Rica. Yeah. And Matt's published over 50 papers, several of them being first author papers, but he's also graduated more than 14 graduate students. And me and Matt, we first met each other at an AGU probably a long time ago, and we've been friends ever since. And Matt's been really supportive of work that we've done, and I'm really excited to hear his latest work here on stable isotopes and isoscapes in New Zealand. So please welcome me, and please welcome Matt, and looking forward to your talk. Thanks, Matt.
Okay.
Take it away.
Okay, thanks. Yeah, it's good to be here. Thanks for the introduction. So I'm mainly a paleoclimatologist, but I'm going to talk about something that's a little bit of a side project to that paleoclimatology. After a while, I got bored of talking about speleothems, and so I thought I'd do something different. And I guess several of you are in the stable isotopes class as well too. So I'm going to talk about some ideas that help us understand stable isotope systematics, and how we can interpret those data for looking at paleoclimatology. In most of the locations that I've worked, we do sampling of rainfall, surface waters, et cetera, to understand the stable isotope systematics better, so that we can understand the climatic forcings that are going into controlling our proxy records. And specifically, I'm working on cave speleothems, which preserve oxygen isotope variation. So I've done a lot of these little side projects as support for the paleoclimatology, but we actually wrote a paper on New Zealand from sampling that we did that came out this year. So I'm going to walk through some of that. Is this changing here? No.
You may just want to click on the PowerPoint.
Yeah, there we go. So I'm actually going to show you the results of this paper first, and this is a map of the South Island of New Zealand. This is an isoscape. So an isoscape is a map of the stable isotopic compositions. And I'm going to tell you how we got to the creating this map. So this is collaboration with Chris Moy, Christina Riesemann. They're both down in University of Otago in New Zealand. Andrew Laurie from Niwa in New Zealand, and then Haroon Stephen, who's an engineer at UNLV, who's worked a lot with the GIS data. So I'm going to show you some photographs as well of the types of you got logged in from another device. Your polling session is inactive. Maybe that has something to do with it. There you go. Yeah. Okay. Okay, cool. So the question is, why isoscapes? And this is where we get into the stable isotope geochemistry a little bit. So isoscapes are maps of a stable isotopic composition. So we're looking at space, the variations over space. And specifically why I'm interested in them is because paleoclimatic records that are isotopic in nature use stable isotopes, to infer climate variations. And for example, we use lake sediments, speleothems, ice cores, et cetera, and those stable isotope ratios are encoding climate information. Okay, so they're recording these variations in climate. But producing these stable isotope maps helps us use this data to interpret our paleoclimate records. And we can also do really interesting things like stable isotope forensics, like fingerprinting, where people have moved from space to space based on stable isotopes in their hair, or provenancing wines, or anything like that, where you can use stable isotopes to pin down a geographic origin of something. But it's also really useful for looking at climate dynamics. And also what we really want to get to in the terms of the paleoclimate would be climate forcing. So just some examples, we've got lake cores happening in New Zealand, cave records from New Zealand. I'm not working in paleoclimate in New Zealand, but I'm helping out these other researchers there. So let me talk about what the objectives of this type of work are. Our goal is to use essentially the spatial variations in stable isotopes as a stand-in for the climate forcings. Typically, what will happen in understanding stable isotopes is you'll look at how they change over time. You might measure rainfall, stable isotopes, and see how climate is controlling those. That's nice. That's a great way to do it, but we can also look at those variations over space and constrain them to the climate fields that they're occurring against. And the nice thing about this is that it allows us to quantify some of these stable isotope climate relationships. And what we're going to be doing here is developing this Isoscape so we can help interpret and select sites better for paleoclimatic analysis. But more, I think importantly, we can come up with a statistical model that relates climate variations with the stable isotope variations so we can actually have predictive power. And that's something that's often lacking in many of the types of works. Now, before I get into what works, I'm going to tell you all the things that the reviewers told me will not work. I spent 25% of the time on this paper learning about how nature works and 75% of the time doing tests to satisfy the reviewers. And this is a list of all the reasons why this approach will not work, right? If you look at an isoscape, you're just looking at snapshots in time. A snapshot in time is like taking a photograph. You might capture an anomalous event, but it might not give you the whole picture. You're mixing, the sample period and correlating it to like mean annual climate variations. So you're mixing times. You're mixing, you're not taking into account seasonality, for example. Like maybe there's seasonal variations in the stable isotopes, and that's going to completely mess up everything that you do. There's an unclear relationship between how rivers and rainfall isotopes are related. We're collecting at a particular spot in space, but that water came from higher up in a watershed, right? And so we have this mismatch in space. We've got weather potentially affecting this, and it's not a climate signal at all. This is the biggest thing the reviewer said. This is never going to work. And we have different ages of the water. Like you collect a river water sample, you don't know what the age of the water is. It didn't form the minute you collected it. It could be a year, a couple years, even older if it's getting groundwater input. And all these things could complicate the story. You might have groundwater, for example. Okay, so there's a whole list of reasons why this approach will not work, and you shouldn't waste your time on it. But I'm going to show you why it does work, and it actually works really well and better than we had expected, even given all of these limitations. Okay, now we're back again to this issue. Now what? There we go. What do you do differently?
I just put there, you said there.
Oh, okay. Maybe that's what it is. Okay. So I'll just give one example of how we can use this information. So this is a cave record actually from the North Island of New Zealand, and this is the oxygen isotope curve over the last 10,000 years. And essentially what we can see is that there's a long-term trend of the stable isotope data, and it's explained relatively well by modeled sea surface temperatures in the Tasman Sea. Okay, but the connection, how you go from SST in the Tasman Sea to a speleothem record for Delta 18O, there hasn't been a whole lot of work yet been done in New Zealand to try to quantify these relationships to explain what the dynamics potentially could be. So I'm not going to really talk about the paleoclimate, but this is the implications for the type of work that we do. Okay, so for those in stable isotope geochemistry, stable isotopes 101, just to remind you, we're talking about delta values. So delta 18O values, where the delta just means in our case, the 18O to 16O ratio of a sample relative to a standard, right? So we're looking at those deviations from there. And then that's because most of the water is H216O, but we're looking at that rare stable isotope H218O as well too. And so I'm going to be just talking about the delta 18O here of these samples. Now, we measure all of these relative to some sort of reference material, and usually that reference material is SMO, which is standard mean ocean water. That's our scale that we use. And then we have a couple of other standards. This is the Greenland Ice Sheet Project, and then standard light Antarctic precipitation. So these are our reference samples for calibrating our delta 18O value. So all of our analyses are done relative to these reference standards so that we can know the true value. Okay, and now we've run out of most of this, so we're using these USGS water samples as well. Okay, so that's kind of how you do that referencing. So we run these the old school way on a TCEA in the isotope lab at UNLV. Basically, we just pyrolyze the sample at high temperature, splits off the hydrogen from the oxygen combines with carbon, you analyze carbon monoxide gas and H2 gas, and then you can get the isotopic ratios of those two. So we do this, this might look familiar to some of you who are running continuous flow stable isotopes. Okay, so that's kind of the background of this. Now let's talk about the real reason we're doing this. The real reason is we want to understand the relationship between climate and stable isotopes. So I just call this stable isotope climatology. I would argue that stable isotopes should be one of our key climate parameters, because unlike temperature, unlike rainfall amount, they actually encode climate information along time and also along pathway. So they reveal a lot of extra information about how climate works and how climate dynamics work. And so I think we need to spend a lot of time in the community understanding stable isotope climatology to the best that we can. And of course, the main reason is these variations in stable isotope ratios are reflecting variations in climate. Okay, now again, if you've taken stable isotope geochemistry, you've probably heard about the stable isotope effects. And I'm just going to really briefly go through a couple of these just to give some perspective on the types of work that, the types of information we're trying to pull out of our stable isotope values. And then finally noting that the paleoclimate proxy data, like lake sediments, speleothems, et cetera, they're preserving these variations that are happening in the precipitation, which is linked to the climate shifts. So anything we can do to better understand those processes really helps us. Okay, so this is kind of the classic temperature effect in stable isotope values, and it's just showing mean annual air temperature versus delta 18O. And you see going from the South Pole up into the tropics, you see a pretty linear relationship between delta 18O and temperature. Now, this is a global relationship, however. But basically what this does is it's using space, looking at spatial variations in temperature and the mean delta 18O value of precipitation to come up with this ratio. But it's based on, the South Pole and a whole bunch of tropical stations, et cetera. So you're mixing locations and it doesn't have much predictive power for a particular place on Earth. But basically, you get higher values with higher temperatures and vice versa. So this is just one way that the stable isotopes are encoding this climate information. Another one is what we call the source effect, where you're getting air masses coming from different locations that have different stable isotope values. So in our part of the world here, this is a site from Utah, where you have quite negative delta H values from the high North Pacific, and then higher values coming from the Gulf of Mexico, et cetera. And so the rainfall that's occurring at that spot is going to have varying ratios depending on where the moisture is coming from. So you have these multiple competing effects. We don't necessarily know which one of these effects is going to be most important at a particular spot over time, right? And so you need to constrain that for each particular site. One more so-called effect that I'll highlight here is just seasonality, and this shows that over the course of a year, you can get variations in the delta. This is why what I'm talking about shouldn't work, right? Because you get these variations over time, and we might only sample at one particular point in time, so we can't say much about it, but I'm going to show you why that does work. Now, let's get into isoscapes then, because this is where it starts to get really interesting. An isoscape is taking all of the available information, trying to make maps of the stable isotopic variation. And these are individual sampling stations across the world. And then this is the isoscape map here. Now, you find these kind of global isoscapes, and they're used quite a bit in the paleoclimate literature, particularly if you don't have local information. Somebody will say, well, I'm working right here at this particular spot. Let me figure out what the delta H value is from that spot based on this map. But here's the problem. There's a couple of problems. To make this kind of map, you need decades of data, in many cases, 10 years, maybe longer, and you need to monitor rainfall on a monthly basis for a very long period of time. So it's very costly, very time consuming. But this is where you get the best data, right? But then the next problem is you can see that some of these locations are very sparsely sampled. And there's many locations that don't have these rainfall collecting stations. And in those areas in between, you have to interpolate. You have to essentially make an educated guess at what those values are. And you could be wildly off. So there's a lot of limitations of these global isoscapes that people have been using to provide context for their paleoclimate records. And the other problem is a lot of this interpolation is just map interpolation, like you did in Geology 101 or creating contour maps. It's not necessarily physically based and linked to a climate parameter. Or it might just be geographically based, latitude and longitude. Those are not climate parameters, but they might substitute for them. So what we do in our type of work is we try to avoid this, and we actually link our isotope values to specific climate parameters so that we can develop a statistical model that links those two directly rather than relying on some sort of interpolated map, and that interpolated map might have errors. So let me just give you a good example. Here's one of these interpolated isoscapes for New Zealand, and this is where we work. So you can kind of read off of this that we should expect to see delta H values, let's just say around -6 to about -10 per mil. And they're pretty homogeneous over most of the South Island, right? And they came up with this map on a very simple statistical model that's just related to latitude, longitude, and altitude. Those aren't climate parameters. They're related to climate, but they're not climate parameters. And so you can go to this website, waterisotopes.org, which is a great spot to get a first estimate. But it turns out in this area, it's wildly wrong, right? Because they don't have enough input data to create this map. So the better way to do it is you collect rainfall in different locations, and then you can create an isoscape based on the long-term rainfall network. And this has been done for New Zealand. But I think there's only a total of 10 stations in the entire South Island. And the other problem is these groups have never made their product publicly available. I'm not sure why, but if you wanted to work with this as a scientist, you can't do it, right? It's not available. All you have is this figure right here. And then the other problem is they're missing big areas. Essentially everywhere in between, they don't have samples to constrain their model. So again, it has to be interpolated based on these kind of limited physical models that don't necessarily encode all of the climate information. So this is great data. This is where you need to go. And if you have a paleoclimate record that's located next to one of these, you're in good shape. But what if you want to know what's happening in one of these sparsely populated regions of your study area? You might not have that data. So what we do to get around that is we create new stable isotope maps. And what we use are rivers. So instead of sampling rainfall on a monthly basis for 20 or 30 years, we collect spatial samples from an entirety of a location, essentially a snapshot in time, and then look at the relationships between the stable isotopes and the climate parameters over space, because they vary quite a bit over space. Okay, so for this particular study, we analyze delta H&O and delta D on river waters across the South Island. Basically, it's a bucket and rope approach, right? So this is our description of our bucket and rope. And what we would do is drop a bucket down into the fast flowing part of the river, pull out the water sample, put it in a bottle, disinfect everything with lots of Clorox bleach so we don't introduce contamination from one site to another. And then we just drive and we drive and we drive and we drive and we sample and we sample and we eat New Zealand pies until finally, we end up with this very large data set of rivers that are essentially a snapshot in time. Now, what we did though to help this work is we focused on really small rivers, and the idea behind that is small rivers are going to be a more local signal. If you sample the Mississippi River at the outlet, that has nothing to do with the climate in New Orleans, right? So what we're doing is focusing on smaller catchments that should have a more local climate signal, so that when we correlate to the climate variables, it's a more direct comparison. What that means is we avoided all of these super beautiful, huge rivers in New Zealand. We just drove right over them and kept on going. And we focused on these tiny little creeks, small little water bodies. And then we did a multiple regression against the climate fields. And we did an uncertainty analysis and tried to validate this model to see how well it worked. Okay, so I'm just going to show you our sample sites. So we ended up with 193 samples. These are all the blue dots over the South Island of New Zealand. The precipitation monitoring stations, again, are in green here. So over 16 days, we drove 5,500 kilometers. And that roughly translated to one sample like every 34 kilometers of driving. And I was saying at lunch, I have no memory of like New Zealand. Like, ask me about a city there, I don't know, because we went so fast and visited so many places in so little time, I can hardly remember anything. But we have very good coverage, and we have much denser coverage than what you get for the rainfall. So you can see there's a handful of rainfall types, but we have these, full transects all the way across the island. So if there are kind of finer scale variations, we'll be able to pick them up in a way that the rainfall sampling will not. The downside is we're just dealing with a snapshot in time, but we've got great spatial coverage. And it's the opposite for the rainfall. They've got very bad spatial coverage, but they've got great temporal resolution. So it's kind of a trade-off, right? I'm not saying one is better than the other. If anything's better, it's the rainfall. But sometimes you don't have 30 years to spare, to do this in a new site. And just to give you some idea on the size of the drainage basins, we're looking at relatively small drainage basins. These are some of the larger ones through there. But some of these are very small, and so they should have a very local signal. And I would say this is the largest river, the largest drainage basin we included in that entire study. But most of the times there are small little creeks like this. I'm going to walk through a couple of the different sampling sites. We would basically drive to a river, drop a bucket down, take a GPS, get moving. You have all these cool names: Roaring Swine Creek, that's at a delta H&O value of -7 per mil. Here's the Fox Glacier draining out of the high southern Alps. That's only -10 per mil. I don't remember which river this is, but it's pretty. Again, more closer to the Alps. That's Christina Rieselmann. You have to trespass, on farmers' fields, and there's the famous sheep. This one's saying, "Hey, what's going on?" But we walked over and sampled that river where we could. This is the outlet to the Tasman River, the Tasman Glacier. Again, it's only -10 per mil, so pretty heavy values for a glacier. Greenland is -25, Antarctica is -55. So these are really heavy glaciers in terms of stable isotopes. Here we are on the Tasman Sea Slope, where it's the rainforest. We even sampled a spring water at the brewery in Otago. Figure why not? -6 per mil. That's Chris Moy. Okay, so let's try to make sense of it all. We have 193 samples spread out all over the South Island of New Zealand. What do we learn from it? Like, that's the goal here, right? So we want to go from this, all these samples jumbled up, to some nice and orderly interpretation, and understanding of what those samples mean. Okay, so let me show you the real data. So all of our sample sites are color-coded to the delta 18O values. The reds are lower values, the blues are higher values. And so you can see there's a real clear pattern along the Tasman Sea coast of more blue values. So most of the values were like -4 per mil to -7 per mil on the Tasman Sea coast. And then in this kind of the center of the island is where you get more of the reds. Those are down to -12, -13 per mil. And then the values are intermediate, mostly on the Pacific coast, which is somewhere around -6 to -7 to -8 per mil, something like that. Okay, so we have this nice spatial variation, but still, that doesn't allow us to figure out what's going on, for example, in between. And you can see, just kind of a curious note, the only place we didn't sample was right around here. And the reason is on our second day of the sampling transect, they had a huge earthquake, the Kaikoura earthquake, and all the roads got closed. So this is the one place we weren't able to sample, but we drove on every other road basically on the South Island of New Zealand, collecting these samples. Now these tiny small dots are climate stations. And this is where the climate data comes from that we end up using to correlate to our stable isotope values. So we've got really good coverage of the climate stations there as well too. I'll show you how we did that in a bit. But first, let me just show you some of the data. Typically, we plot oxygen versus hydrogen isotopes in something called a meteoric waterline. The meteoric waterline tells us whether there's been evaporation of the samples, and it shows you the relationship between the two isotopes. And the reason I point this out is these are all of our samples. The blue line is our statistical fit, our local meteoric waterline from New Zealand. And then the red dashed line, which you can't see, is underneath the blue. So our samples from New Zealand are exactly the same relationship as what's called the global meteoric waterline. We have the same, the same slopes and essentially the same intercepts in there. So the first thing we can do is we can step back and say these water samples are likely to from the rivers are likely to represent precipitation. They haven't been evaporated, they haven't been altered. They're very equilibrium samples associated with rainfall. And that was important to show that the rivers are not evaporating and changing the isotopic values, which would mess the whole thing up. So this establishes that rivers in a humid environment are good stand-ins for rainfall. Okay, so let's look at a little bit more of the data. We probably talked about altitude effects in stable isotope geochemistry. We see that too. But we have to separate out the different basins. So the red is the Pacific, the blue is the Tasman Sea. When we first plotted this together, we said, oh, there's not much of a great, altitude effect in the stable isotopes. But once you separate out by basin, you can see that they both have the same altitude effect, the same slopes, but they're offset by a couple of per mil. And the reasons are important for what we're going to talk about next. Basically, the Pacific slope has delta 18O values that are about 2 per mil lower than the Tasman slope. Okay, so this was able to demonstrate for the first time that there's a pretty strong altitude effect in the South Island, and that's not a super unusual find, but now we can quantify it better than we've been able to before because we haven't had this data until now. Okay, here's the big question though. What controls the delta 18O values in terms of a climate parameter? This is where we started to look at the climate data. So we have all these samples, we have a latitude and a longitude and an altitude, we know where they come from in space. So what we did is we assembled a whole bunch of different climate variables. We ended up with 117 different climate variables. And we correlated our delta 18O values that we measured to those 117 different climate variables to see where the strongest correlations were, which we could then infer was the controlling force on these delta 18O variations. So just for example, we pulled in what's called the WorldClim database, the BioClim database, and this has things like annual mean temperature, precipitation, mean diurnal range, isothermality, and then we pulled in monthly values for surface radiation, wind velocity, all kinds of different things. Every data set we could get, global aridity, PET values, effective evapotranspiration values, et cetera. So we have those data for every grid spot where we've collected this data. Okay, so that gives us the ability then to develop a statistical model and see what shakes out in terms of the potential relationships between climate and the delta 18O values. And of course, we also had sample altitudes. We calculated distance from the coast. We have latitude and longitude, just from the GPS as well too. So we threw those variables in. Okay, what does that mean from a practical perspective? So here are two maps on the right. Those are for two different climate parameters. This is temperature seasonality. And then on the bottom is precipitation of the driest month. Okay, just for example, these are what the climate fields look like. And then what we did is we interpolated onto our sample locations. So every sample location then ended up with a delta 18O value, a delta D value, and 117 different climate parameters. So we end up with this really big matrix that we can run correlations on. And then this is just some examples of how we run the correlations between delta 18O. So let's pick one that works. So delta 18O versus mean diurnal range has a nice correlation. Nice correlation of delta 18O at minimum temperature of the coldest month. And then some have no relationships whatsoever. So I can't read all these. Precipitation of the coldest quarter, for example, doesn't have a good correlation with delta 18O. So this allows us to pull out the factors that are most relevant for controlling the spatial variations in the stable isotopes. And then we can eventually build a model that will allow us to predict the delta 18O value at any grid point in the South Island and New Zealand and potentially extrapolate it elsewhere. So again, you end up with a correlation matrix, right? So here's 117 times 117 correlation matrix, and the colors represent the correlations. And then delta 18O is this thin little one off to the left here. But just to show you, we did the full suite of statistics on this to try to tease out the most relevant climate parameters. And what are they? Okay, what are the main controls? I'll just give you a couple examples here. So we take those 117 variables and reduce it down to just a few that are important. Our final model, I just chose 4 because 4 seemed like a good number. We could have had more, but we certainly don't need all 117 of them. But let's look at the data on the right first. So this is the map of the South Island, and I'm just showing you a transect of data basically from north to south. So we start from the Tasman Sea, go to the Pacific coast. The bottom shows the altitude. So here's the Southern Alps. And then we have a bunch of other mountain ranges that we cross. And then we get to the Pacific Ocean. So this is the latitude or the altitude transect across the South Island. And then these are our delta 18O values in blue. So they start off at around -5 per mil, decrease to -12, and then increase again to like -6 or -7 per mil. So you see a really clear variation and the delta HNO values on a spatial basis. So here's a couple of different climate parameters. Minimum July temperature shows that same pattern. Potential evapotranspiration in December shows that same pattern. The temperature annual range shows the same pattern. So what we were able to kind of pull out of this is that the parameters that are most strongly related to the stable isotopes are indicators of continentality. So the farther away from the coastlines you are, the colder the winter temperatures. So July, of course, is winter in the southern hemisphere. So you get colder winter temperatures farther away from the coast. In the summer, the potential evapotranspiration is the greatest because they're the most arid locations, because they're continental. Temperature annual range is also greatest. I have it plotted on a reverse axis. And they're also greatest kind of in the center of the island where you're most distant from the coast. Okay, so now we can actually link the stable isotopes to a particular climate parameter, or 117 in this case, or just pick the ones that have the strongest correlations. So what that allows us to do is we take our data points with a point in time, and we correlate them to all these different parameters, including seasonal. So here is a plot of delta HNO against monthly parameters. So I'm just going to focus on this middle one for a second here. So this is our delta HNO values plotted against, these are correlation coefficients for T max, so max temperature, average temperature, and minimum temperature, and on a monthly basis. Okay, so we collected our samples in November, but we're correlating it to every month's average climate values. And we get correlations that are as high as 0.8. We're mixing timescales here. We're talking November 2016 versus average climate conditions, but we still get a correlation that's 0.8. It's a really strong correlation when it comes to climate. So what we're able to conclude from this is that it's the winter temperatures that are, it's winter parameters in addition to the continentality parameters that are the strongest controls on the spatial variations in these stable isotopes. That gives us some idea that it's winter that's most important, not summer, right? So summer has really weak correlations. Summer being, correlations between minus 0.2 and maybe 0.35 or something. So they're pretty weak, but we get really strong correlations. The other thing this allows us to do is quantify the slope of that relationship. So this is the variation in per mil per degree Celsius. In other words, the temperature gradient. The temperature gradient is in the winter somewhere between 0.5 and 0.6 per mil per degree Celsius. And then during the summers, you have really big error bars because it doesn't work very well. Again, it's more evidence that these river water samples are recording winter climate rather than summer climate. Okay, so the reviewers had all these reasons why it didn't work, but this is our final model right here. This is modeled delta HNO versus measured delta HNO. So we see a really strong correlation. What are the parameters that went into this final model? Well, we just really picked 4, and that was an arbitrary decision because I didn't want to have too many collinear variables. And so we do this multiple regression. Essentially, it picks the strongest predictors of delta HNO, and then the next strongest predictor that's not collinear with the first one. So like I could pick July temperature and then June temperature and then August temperature, and they would all fit into this model too, but they're all collinear because they're all related to each other. But this approach avoids that. So in other words, the first we chose was minimum temperature of the coldest month and then December evapotranspiration. Those two are not very strongly correlated to each other, so they're not providing redundant data to this model. And so you kind of go weaker, weaker, weaker in your variables, so long as they're all non-collinear. And then we will correlate it to surface radiation, and then the final variable was latitude. And these were picked by the model and the regress function in MATLAB. But I could have added more parameters. I could have picked my own and tried to improve it, but I didn't want to do that. I wanted to just limit it to a small number of variables to get the best model. And then this is what we ended up with. So in other words, we can predict pretty well the delta H&O values over space with this simple model. It's just got 4 climate parameters in there. Just to give you an idea of how good the fit is, this is an R value of 0.88, but of course, it's being measured against itself. Like we're measuring it against our own data, so it's not really a true model test yet. We'll come back to that a little bit later. So then we can do like an uncertainty analysis, like how wrong could we be if we use this model to randomly select our own samples. So we kind of took a jackknife approach. What that means is you run the regression by randomly withholding a certain number of data sets, a certain number of samples. So we ran this 1,000 times and then randomly excluded, what was it, 20% of our samples. And then you can look at the root mean square error, which is how far away you are from your regression line, to figure out how well that model performs based on the input data. So in other words, we can predict from our model our own values within 1 per mil, and this is rounded up, right? So 1 per mil or so, and most of the residuals here we're within 1 per mil of uncertainty. Again, this is measured against our own values, so it's not a true test. It's just it's an estimate of how good that model is working based on the input data. We need to validate this against other independent data sets to get a better idea of how well it works. Okay, so then we take that simple regression model. It's just got 4 parameters, but we have four climate fields. We have all these maps of the different climate fields, and we just come up with our, I forgot to mention it, where's the equation? Yeah, here's our equation right here. So all we need to do is take the November surface radiation map and the December evapotranspiration map and the minimum temperature of the coldest month map and a map of latitude. We just run them through this equation and then we end up with this isoscape, right? So this is what I showed you in the first slide. So this is our multiple regression model isoscape based on those 4 input parameters. And the nice thing about this is it interpolates, well, it's not really interpolating, it's a physically based estimate for every grid point in that whole map. We don't rely on interpolation. Like each one of these data points has climate as the predictors. So that gives us a lot more power because we can say at any particular spot, we can calculate what those different climate parameters are. Or we know what they are, and we know what the delta H&O value should be at that spot. Okay, so what do we see? We see this kind of broad Tasman Sea blue stripe of high delta H&O values. And we see this really big red spot right in here is what I'm calling the isotopic rain shadow. And these are where the values get down to -11, -12 per mil. The stripe of the Southern Alps, the high spot of the Southern Alps is right where I have that text essentially. The lowest values are not at the highest altitudes in the Alps. They're in this inland region and extends almost all the way to the Pacific coast of really, really low delta H&O values. And the reason is this is the area where you've got the most continentality and all those other different predictor variables are really strongly expressed. So one of the surprises from this is that where we had expected to see the lowest delta H&O values would have been like on the top of the high mountains where you've got the big glaciers. They weren't. They were like -10 per mil. But once you go into these small little streams in central Otago, you get -12 per mil, -11 per mil. And so it's not altitude. It's not a direct temperature effect, on that. It's related to the overall climate configuration of the South Island, which is limited or which is controlled by that continentality. Okay, so this is a physically based model. It's not interpolated. We can do a much better job at estimating delta H&O values for paleoclimate locations or working on anything related to stable isotopes that's related to fresh water than if we use these precipitation maps that were interpolated where they didn't have data. So what does this kind of mean in terms of a climatic interpretation? Essentially what we have here is we've got the westerlies coming from the Tasman Sea. They are bringing the least fractionated moisture that's highest delta H&O values. It starts to rain out along the Southern Alps. So you start to remove the moisture due to Rayleigh distillation, where the heavier isotopes are progressively falling out, leaving the residual air mass more negative and more negative. Until finally you get into this area here, where almost all of the moisture is likely coming from the Tasman Sea, and it's already been isotopically distilled pretty significantly by the time it reaches there. Even though it's not the coldest locations are up on the top of the Alps here, but the lowest values are in the lee of the Southern Alps, because they've experienced all this rain out as they got lifted up over the mountains. And then we have the Pacific Ocean, where there's actually some easterlies coming in, and you can see that there's a thin strip of higher delta H&O values along the coast as well too. So we have this combination of easterlies coming in, contributing less fractionated moisture, the Tasman Sea coming in, contributing less fractionation moisture on there, and then in the middle, this isotopic rain shadow of really, really low delta H&O values. Okay, so we've learned quite a bit from this. We've learned that if you want to study Pacific Ocean signal, like anywhere down here is pretty good. If you want a pure Tasman signal, pick your spot. It goes all the way up here. And if you're interested in studying a paleoclimate signal where you should have the most extreme values, you pick it from the isotopic hole in here. So we can look at this just in a topographic profile like I showed you before. So the westerlies are starting off high values, you get Rayleigh distillation until you end up with this rain shadow right here with the lowest delta H&O values. And that's kind of in this area here, because you have blocking from all these mountain ranges that blocks the Pacific moisture. It can't get in there. The Tasman moisture comes up and over, and then by the time it gets on the inside, it's really isotopically negative at low values. Notice where the lowest values are. They're not on the top of the Southern Alps like you would expect. They're in this interior region here. And that was a bit of a surprise to us. Okay, so what did we learn out of all of this? The river delta H&O values are strongly correlated to winter temperatures and continentality. And continentality and winter temperatures are both linked as well too. The lowest values are not from the high mountains or the glaciers. They're from these tiny little creeks, tiny small water bodies, streams in the interior of the island. We have this really strong isotopic rain shadow where we have essentially 8 per mil decrease over the span of 50 kilometers or so. And that's a huge gradient. When it comes to climatic control, 8 per mil over roughly 50 kilometers is really, really big. And it shows you that the Southern Alps are really powerful climatic forces. They're causing all this rain out to happen and have a decrease in the delta H&O values. The Tasman slope really has a pure Tasman signature. You're not going to get Pacific moisture over there. The Southern Alps are too high and the stable isotopes show that. But once you get into the interior, you get a mix. You get a mix of Tasman, and as you get closer and closer to the Pacific, you get a mix of Pacific plus Tasman. So what that means, if you're studying a paleoclimate location right over here, it's kind of hard to disentangle what the isotopic value is coming from the west versus coming from the east. You've got a mixed signal. So if you want the purest signal, you go somewhere to the west here or directly on the Pacific coast where there's not much Tasman moisture making it. I didn't really talk about too much about the moisture source variations, but we see that in deuterium excess as well too. Okay, so finally, how do we apply this to the paleoclimate data? Well, if you're working in an open lake system where it's maybe being fed by river water, it's not a closed basin, water's flowing through there, winter is the dominant control in terms of the delta H&O values. And we can also quantify that relationship between delta H&O and temperature based on what we see in our correlations. So you have a better control over how the stable isotope variations in your paleoclimate proxy are varying relative to those climate parameters, based on this statistical model. Now, how well does this actually work? Okay, I want to show you two maps. This is from that simple model from latitude, longitude, and altitude. And then our model right here, the color ramps are a little bit different, obviously. But you can see we've got this really strongly expressed rain shadow right here. It doesn't even show up on that map. Not to say there's a problem with their techniques, but they didn't have enough input data to create a map of an isoscape that was sufficiently resolved to show what's really happening on the ground. This technique of creating surface water isoscapes allows you to fill in these details on a very short period of time, and you can see more climatic information with a 2-week or 3-week sampling campaign than you can do with several years of precipitation monitoring, right? So this just doesn't even pick it up. That's kind of the lesson from that particular comparison. How wrong is it? So I just took our most negative value, minus 12.8 per mil. We measured it. This model suggests it should be minus 9.5 per mil. So it's off by 3 per mil out of a total variation of roughly 8. So, it's like 40% wrong. And then our isoscape model, of course, we use it as input too, but our isoscape predicts it within 1/10 of a per mil. So you don't even want to use this map if you're working in New Zealand. It's not useful. It doesn't have enough information. But this map is quite a bit better because they do have more sampling stations. This is from the precipitation samplings that were done by a different group. The color ramps are reversed in these two. But they do see this isotopic rain shadow in the blues here, and we see it in the reds. But ours is much more resolved because we have much higher resolution sampling, and we have higher resolution map. So we interpret. I keep on using interpolate. We calculated it based on a much finer spatial scale, which is roughly 30 arc seconds. So it's a really high resolution map. The other thing is our model is publicly available. Anybody can use it. Their model is not publicly available, so if you hold on to the data and you don't share it with anybody, it limits the utility. I don't know why they don't share it, but they don't. But you could also argue that they're kind of missing the structure here because of their coarse sampling resolution and their coarse map resolution. They're not really seeing the full picture of what's happening with just the precipitation solutions. Okay, so here's why I say this works. This has a lot of strengths. It has weaknesses too, this approach. Ideally, you're going to do both if you can. Rainfall monitoring and the river water isoscapes. But you can do much more dense sampling from the rivers than you can with precip. And you can have, because of that denser sampling, you pick up more detail that's missed in other records. And then it allows us to look at things like the topography, the development of the Alps in much more detail than you could with just a few precipitation sampling stations around. Okay, so we also did some model validation. Like, how well does this work if we test our model against totally independently collected data? Because this is what the reviewers made me spend 2 years doing. That's how long it took. It took me 2 years to do all these tests that they asked for, and they were good to ask for it, I guess, because now I felt more confidence in it. But we looked at this old data. We scrubbed a whole bunch of data from the literature. In every case, we were able to predict delta H&O values of rivers collected in the 1980s within 1/10 of a per mil, plus or minus 0. We'll just call it, that's the uncertainty. So let's call it 1 per mil. Right, so these are old river samples. Then we tested it against 127 other river samples, and we were able to predict these other river samples, again, totally different workers, different time periods, but we just figured out the delta H&O values from our model compared to theirs. Again, we were within 1 per mil of their values. And some of these were collected 20, 30 years ago. So we can do this really well. This is just a plot of, our predicted versus our measured, when looking at these independent data sets. Not perfect, but I'd say within 1 per mil is pretty good out of a range of 8 per mil or 9 per mil, something like that. Let's see what else. And we also compared it to rainfall. So we used that rainfall data as well too. And we found out that our river values are strongly correlated to winter rainfall, basically the 6 months prior to November. And then we get also a really good measurement versus our prediction. So this model even predicts the rainfall values, even though it's not rainfall, they're based on rivers. So it's still a pretty good fit. So it works. It's not so complicated as we were, suggested that it would be. Okay. What are some of the broader impacts finally? Let's just kind of end on this. Once you have one of these maps, you can do lots of things with it. You can make predictions for other systems. So this is an isoscape based on our river waters, but just making the assumption that, let's say the cave carbonates are forming from waters of that same isotopic composition. You take the temperature map and the isoscape map, and we can make a prediction for what the delta H&O value of calcite in a cave will be at any particular spot in New Zealand. There's not caves everywhere, but it's a prediction, right? So we can make a prediction of this is speleothem calcite delta H&O values based on an equilibrium value. Or we can do things, we can also create the hydrogen isotope, map too. In this case, we didn't run the full regression. We just used our water line. And so this is a prediction of the delta deuterium values for river waters at every grid cell over this area. And this is, of course, useful, the type of thing that Jess does, looking at leaf waxes, et cetera, that are recording the delta D values. Some other implications of this, some of you might have heard of the work that's been done looking at the uplift of the Southern Alps. Basically, the idea is that the Southern Alps have been uplifted over since the Pliocene, and that isotopic rain shadow has developed since the Pliocene. And they used kaolinite clays to try to show that over time, the kaolinite clays were getting more and more negative. And it turns out they sampled right there to look at the kaolinite clays since the Pliocene, and they picked exactly the right spot, right in that isotopic rain shadow. And they didn't really know it, because they did this work a while ago. So we can predict the delta H&O values of kaolinites, making these same assumptions, which might be wrong. We can calculate feather deuterium isotope values. Like here's the national bird of New Zealand, the kiwi. If you get a kiwi feather, you can figure out if it came from the Tasman slope or the isotopic rain shadow based on measuring the hydrogen isotopic values and our isoscape. So this is bioforensics. You can use this to fingerprint. Here's an isoscape for sheep's wool. Basically, you can find the equation, you can make the isoscape, right? So I could predict sheep's wool is going to be around minus 60 per mil right there, and the sheep over here, sheep up here are going to be maybe minus 10 per mil. You can provenance that and figure out if somebody's selling counterfeit wool. If they're saying it came from New Zealand, but it came from Austria or something. We can do this for human hair. We can figure out if people are migrating from here to there, or migrating back and forth across the island, because your hair is going to encode that same isotopic variation of the water that you ingest. Or we can do it for tooth carbonate or even bone phosphate, because there are relatively simple equations that relate delta H&O to these different parameters. So there's all kinds of broader impacts of this type of work where you can use it for archaeology, bioforensic, paleoclimatology, et cetera, and they give you the ability to make predictions. So you come back to the speleothem record here, and it kind of fits. The interpretation of the speleothem record from New Zealand as an SST control in the Tasman Sea. Well, that makes total sense because temperature is the strongest control, on the stable isotope variations in our data set as well, too. And they made this conclusion based on modeling data, but it also fits with the information that we have as well too. Let me just end on one last slide. Is because this model is physically based, we can use it for projections into the future. If temperatures warm, for example, during the winter, our model is going to predict that the delta HNO values and the delta deuterium values are also going to increase, and by a known amount. So in 100 years, we can test to see if this model is junk or not. I won't be around. Okay. And so this gives us predictability, and that's the really nice thing about this type of model. Okay, so I think I've mostly used my time, so I'm just going to end here. This is totally unfunded work. We've never been funded for doing this. I've never even asked for funding. It's just been a fun kind of side project that we do. And then I do want to thank all those critical reviewers, though, because they helped us understand the system. And even though it took an extra 2 years to get this paper out, I think it was worth it because they were so harsh on us, at least the first time. And all this data is publicly available. So anybody can download it, use it for free for whatever you want to do. All right, thank you.
We've got time for questions. I'm going to turn on the video got turned off. That's okay. So let's take a couple questions maybe from here. Questions?
Yeah.
So the South Island of New Zealand is in this sort of sweet spot with respect to latitude, where the westerlies are coming in, and then from the Pacific Ocean, the easterlies are coming in. So what's the sensitivity of that rain shadow to being in that sweet spot latitudinally, and what happens if those winds shift in latitude?
I think that's a great question, and it's also a great rationale for asking for funding to do another sampling campaign at a time interval when the westerlies were at a different location. You know, for example, if there's a different ENSO mode, or some control on where those westerlies are moving on an interannual basis, repeat the sampling and then see how the isoscape differs to look at how that would happen.
Yeah.
Yeah, because we can't really say that yet. It's a great question, though. Amy.
So I understood your point about wanting to do really small catchments, which makes sense to me. But I guess some of your catchments are also capturing melted glacial water.
Yeah.
And so, I mean, the rationale for the small catchments is kind of a snapshot of equivalent snapshots everywhere. But the glacial water could be representing 100 years of different precipitation. So is that I mean, did you ever just take those out and look at those individually, or look at your data set without those glacier meltwater rivers?
Yeah, we did pull out that data. So I coded each river basin, whether it was glaciarized or not glaciarized, and I repeated this, and we get the same results. And it was actually really interesting. There was very little difference at all. But most of our samples were not glacier fed because we focused on the smaller catchments away from the Southern Alps. So even when we were in the middle of the Southern Alps, you see a glacier over there, I would walk over to the snow patch and get the water draining out of that instead of just the glacier water. So I would say maybe 10 or 15 samples at most had a strong glacial contribution, and then the rest of them did not. Yes.
Since you mentioned that you checked your models against data that you'd pulled from previous literature, out of curiosity, did you try looking at how your model stacks up to data from maybe even further back recorded in speleothems or ice cores, or just to see like how far back in time your model would be accurate?
Now, we didn't use paleoclimate data, partly because there's not a whole lot available. Like there's not enough grid points, to probably have a statistical fit. But it's a good idea. I mean, I should do that. Yeah, I think it would be fun to look at that if we could find it. But you still have to convert it, like a speleothem, you have to convert it using temperature and the fractionation equation back into a water. But there's really not much in terms of caves yet from the South Island. Maybe some of the lake sediments. I'm not sure how much they've done with the hydrogen isotopes yet, but it's possible that we could do that. Yeah.
Somewhat related question. It sounds like I might know the answer because you said the data isn't available, but I'm curious if you have any data to test the space for time assumption. So in other words, I'm thinking about applying this to paleo data, right? That these relationships hold equally through time as they do through space. And I guess if it's continentality, it probably should, but do you have any way to test that with your available data set?
That's a great question. And I haven't done it. Because I'm not aware of the paleoclimatic data that's been quantitatively interpreted as temperature. You know,
I mean, we're just like observational isotope data. Is there enough of a time series that you can test like anywhere from the South Island?
yeah, so one test that we could do would be, for example, looking at the temperature gradients in precipitation versus the temperature gradients in the river waters. I didn't do that directly, but they fit within the range of where we would expect them to be. But I would agree that's a good place to go next. Yeah.
Your data shows a strong correlation with the altitude, but in your final equation you didn't include the altitude. Is there a reason? Because it involves global modulation, they also use the altitude.
It was the model that chose most of those. So what I did is I allowed the regression model to choose the variables itself. And I took the first 4 that it chose, and altitude was not one of them. So I could have put it in myself, but then that would have been introducing my bias into it instead of just letting the statistics decide. And also altitude is collinear with temperature, and there was already a temperature parameter in there. So I think that's why it ended up not being included there. So I think we had minimum temperature, the coldest month is also correlated with altitude. And so they would be bringing redundant information in there.
Temperature could also be correlated with latitude.
Yeah, latitude was one of them in there, but it was very weakly correlated. So there's kind of thresholds that the model uses for picking how strongly 2 different variables can be correlated while still including them. And so it only picks the ones that fit that model. Yeah.
This might be a little bit dark question, but did you sample a catchment where people are also sampling precipitation for like a direct measured comparison between precipitation data and river data? Perhaps there's like a correction, maybe you want to sort of start being like, well, can we compare this to precipitation? Maybe there's like an offset, you can normalize it.
Yeah, the answer is yes, we did sample those catchments, but I didn't pull out the data specifically with that in mind. I just treated everything in aggregate. You know, I took all the precipitation samples versus all of our data samples and then did the comparison. But I could do what you're saying in more detail, yeah, to see how it relates. But my vision was, let's look at the entire South Island rather than an individual catchment, because I wanted to see the bigger relationships rather than the more local ones, because it starts to break down more. Usually the more local you look, but it would be a good place to look next.
Kind of a detailed question, but I noticed your map said residual corrected, and I was just wondering what you meant by that.
Yeah, I didn't talk about it because it would have been really getting into the weeds.
Yeah.
So when we first created our Isoscape map, I was able to calculate the residuals between what our map said and what our measured values were. And so the residual corrected map is, I just interpolated all those residuals and then subtracted them out from the model. So it gave an improved fit for that model.
Does that mean you think the residuals are structured and not random?
So the assumption is that you can do that if the residuals are uncorrelated with the model results. They were correlated with our measurements, but not the model results. But I can also say that it doesn't matter for that map. It looks exactly the same whether we did that residual corrected map or not. Yeah, so it wouldn't change anything in terms of the overall interpretation. but it would change probably the gradient slightly. You know, if we needed to, you know, use those quantitatively.
Matt, one last question here. Curious about the mechanism for why you get these small catchments being so depleted as opposed to altitude, for example, right? So, and obviously evapotranspiration and evaporation was one of the factors that went into your model. So does that mean that these rivers are sitting, not getting much of P, but getting a lot of E, and that's why they're so depleted, or what's going on to cause that depletion?
Well, if they were evaporating, then they would actually have increased values rather than decreased
Right,
values.
that's right, opposite, yeah.
Yeah, and so the first line of evidence is that even those really negative values plotted right on our meteoric waterline, they don't have anomalous deuterium excess values, which I didn't show maps for that, because that would be the proxy for
That's right.
PET.
Yeah.
And they were not correlated to that.
So how do you get them so depleted?
It's local rainfall. So it's also colder in the center. Okay, so you have the Southern Alps, it gets really cold, of course, on the mountains. Those air masses come down, but then they might be, you know, cooling off at nighttime and precipitating out at that point. So you still need to drop the temperature of those air masses, even in the lower altitudes, to force the precipitation. But the incoming moisture was already so negative that the precipitation ends up being really negative, but it doesn't rain that much, like, because it's pretty arid there too. So you just need some drop in temperature to produce that from a really fractionated moisture mass. Yeah.
Okay, another round of applause for our speaker. We're going to be going out for beers at Gentle Ben's if anyone wants to join. So thank you. And by the way, please, if you can just turn your tables, thank you very much. That helps out our meeting.